Global warming is a growing challenge, with melting glaciers, rising sea levels, and more frequent wildfires and droughts as clear consequences. According to the Environmental Protection Agency, a major contributor is methane, a greenhouse gas that traps heat 28 times more effectively than carbon dioxide. Cattle are significant methane emitters, releasing up to 80 million tons annually through anaerobic fermentation. This process, known as methanogenesis, involves specialized microorganisms called methanogens that convert carbon dioxide and hydrogen into methane, which is then released into the environment, mostly through burping. With global demand for red meat expected to double by 2050, reducing methane emissions from cattle is essential. During our brainstorming phase, we came across a promising solution for sustainable cattle farming, which involves using feed additives derived from red seaweed. This seaweed produces bromoform, a compound that inhibits the final step of methanogenesis. Studies show that such additives can reduce methane production in ruminants by up to 98%. However, frequent administration of these additives increases costs for farmers, making it unsustainable for large-scale farming. Introducing iGEM Lund 2024. We are Methane ReMOOver, a team of 15 enthusiastic students from diverse nationalities and academic backgrounds, representing Lund University in Sweden. Our goal is to engineer bacteria that can harness the power of red seaweed to continuously produce bromoform in the cow's rumen. With this approach, we expect to not only solve the issue of frequent administration, but also increase the dietary energy available to the cow for milk and protein production. Ultimately, we aim to offer a long-term, cost-effective solution to reduce global methane emissions and contribute to a more sustainable future.
In our design, we were strongly inspired by the team of Maastricht 2021 and Nebraska 2017, who, as part of their project, expressed the gene bromoperoxidase. In our construct, the bromoperoxidase produces bromoform and other brominated compounds from bromide salts, hydrogen peroxide, and methyl ketones. The hydrogen peroxide, in our case, is produced by an NADPH-dependent oxidase. During the design phase of our project, we consulted with Tom Williams, the CEO of an Australian-based company called No. 8 Bio, who focuses on reducing enteric methane production from cows. He recommended we use the microorganism called Cuprividis necator due to its ability to compete with methanogens for the same substrates, its ease of cultivation, and its ability to survive within the cow's gut. Based on his recommendation, we chose Cuprividis necator as our chassis. Additionally, Paul Hudson, a professor at KTH and expert on C. necator, provided us with valuable guidance on handling the microorganism while also sending us a sample. We selected the low-copy plasmid, PCAT201, to insert our construct. We chose it due to its broad host range and its ability to overcome inefficient transformation through electroporation. As a proof of concept, the bromoperoxidase gene, MBB1 or MBB4 from A. taxiformis and the vanadium-dependent bromoperoxidase gene from Carolina pilulifera is regulated by the ARA-BAD inducible promoter, activated by L-arabinose. A His tag was added for easy purification and isolation of the expressed protein, while the NADPH oxidase gene is driven by a weak constitutive promoter. First, we transformed E. coli with the MT-PCAT201 plasmid. Then, we purified and linearized the plasmid, confirming the digestion via gel electrophoresis. In parallel, we amplified the target genes by PCR in preparation for a Gibson assembly. The assembly was performed in 2 stages. In the first, we inserted the bromoperoxidase gene along with a promoter, and in the second, we added the NADPH oxidase. After obtaining the final construct, we transformed it into E. coli for amplification and verified by colony PCR and sequencing. Finally, it was transformed into C. necator via electroporation before starting with analyses of the protein and its activity. The first way of testing the presence of the bromoperoxidase was by visualizing the protein samples pre- and post-induction via SDS page. A Western blot was also attempted to verify if the identified bands were truly the protein of interest. The halogenating activity of the enzyme was also tested using 2 different approaches. First was the phonation of phenyl red, and the second was the monochlorodimidone assay. Finally, the methane reduction capabilities of the constructs and Cinecator were tested directly in anaerobic sludge using an AMPTS system and gas chromatography.
One of our biggest challenges was the PCR amplification of our fragments. Even though we have put a lot of effort into designing primers and consulted specialists such as our supervisors, real amplifications are far from the ones performed in silico. Hence, in the initial stages of our experiment, we had to devote a lot of time and energy in optimizing our reactions. We performed various types of PCRs. One worth mentioning might be the touchdown PCR. However, still, in the end, 2 of our constructs showed unspecific byproducts. How we resolved it is by giving up work with one of those. However, the second one was special and very unique for our experiment, so we wanted to continue working with that one. Here, we cleaved it out of the gel and performed a gel extraction. So another challenge we have encountered was the vast waste production. During our laboratory activities, we have noticed that we produce large amounts of not only bacterial waste, but also of plastic waste. And because we wanted to be sustainable, we tried to reduce our waste production by following 3 simple rules of recycling, reducing, and reusing. We were trying to reduce our plastic waste consumption by using glass where possible. Next, we tried to reuse a lot of our materials. So for example, these gloves that I have on can be used a second time if I don't perform any dangerous activities in them. And lastly, we are trying to employ recycling where possible. I think our biggest accomplishment might have been the Gibson assembly, because we have envisioned to combine large fragments altogether 6.5 kilobase pairs with our plasmid. And we wanted to perform all of this in one go. And we weren't sure if this will work, so we had also backup strategies. However, after the assembly and also colony growth, we could conclude based on the PCR and sequencing result that the assembly succeeded. We were successful in assembling our plasmids, carrying the promoter and the vanadium-dependent bromoperoxidases. However, after induction, only one construct showed promising results with protein synthesis. Upon further tests of this sample, we sadly could not detect any measurable protein activity. Further plasmid sets, carrying additionally the NADPH oxidases, showed also successful integration. However, sequencing results showed mutation in the coding sequence, and our efforts at correcting these were unsuccessful. Currently, as we are speaking, we have a co-culture between methanogens and our most promising samples ongoing. We are hoping for successful results.
Our project design prioritizes safety at all stages. All components are on iGEM's whitelist. The team received safety training, the lab is equipped with necessary safety tools, and we work in team to ensure safety. While bromoform is toxic, the study shows only trace amount in cow's urine and milk. The concentrations in milk are well below the safety limit of WHO. The antibiotic waste is autoclaved and disposed of properly. The non-antibiotic waste is autoclaved and disposed as biowaste. Our bacteria samples producing bromoform will be kept in the film hood to evaporate overnight.
We focus on the understanding of the bromoperoxidase mechanism by adapting the Michaelis-Menten to fit the BP ping-pong mechanism. We use tools to predict the Km and the Kcat for MB1 and Carolina, and finally simulate the reaction dynamics. Structural analysis was conducted through sequence alignment and catalytic site identification for MB1, MB4, and Carolina. And then AlphaFold was used to predict the structure of MB1 and MB4, also to estimate how syncope mutation affect the catalytic activity. Additionally, a Hill-like equation helped estimate enzyme production by varying factors such as promoters, ribosome binding sites, and plasmid copy numbers. These enable us to predict the induction point, protein levels, and gene expression. Lastly, we modify a GMO-scale metabolic model of Cinecator to include the reaction catalyzed by the bromoperoxidase and the NADPH oxidase. This model revealed that the organism prioritized product formation over rapid growth, aligned with our goal to use it as a probiotic. We also modeled the competition between Cinecator and methanogens by merging genome-scale metabolic models of both organisms. These allow substrate competition between them, revealing that
Sea nectar has higher growing rate compared to methanogens.
Throughout our project, we actively incorporated feedback from key stakeholders to ensure that our approach aligns well with their expectations and real-world applications. We reached out to LRF, an organization dedicated to supporting the interest of farmers and agricultural businesses, and spoke with a specialist there. She noted that sustainability is a significant challenge and a product that reduces methane emissions could provide much-needed relief for the farmers. We interviewed 2 farmers about their views of synthetic biology and openness to trying new technologies in cattle farming. Although initially skeptical, they expressed willingness to try methane-reducing products as long as they prove beneficial for the cow's health. During our visit to Bolerup farm, we explored how our probiotic could be added into the cow's diets. We found out that the farm is highly automated. Feeding and milking are managed by automated machines, adjusting the concentrate mix based on each cow's milk production. In theory, our probiotic could be added into the concentrate mix, ensuring precise dosage for each cow.
During our project, we connected with other iGEM teams and organized the Swedish iGEM conference, where teams presented their project and discussed, among other subjects, human practices and education. To further engage the younger audience in synthetic biology, we proposed developing a game. Inspired by previous iGEM games, all the Swedish teams collaboratively created Plasmid Assembler, a printable card game that you can find on our wiki, designed to make learning about synthetic biology fun and engaging. One part of our educational outreach was to educate teenagers about synthetic biology through workshops and school visits. We delivered lectures on synthetic biology, gave insights on our project, and gave students the opportunity to design their own plasmids. Students got the chance to explore this field and learn about the various ways that science can offer solutions to make this world a better place. Our team also participated in this year's Kulturnatten, an annual culture event featuring art and science all across Lund. We focused on educating people of all ages about synthetic biology and our project. We also had some fun activities such as pipette practice and introduction of our game.
In 2015, all UN member nations adopted the 17 Sustainable Development Goals to drive global progress by year 2030. Our project aligns closely with several of these goals, and we have focused on 3 of them: climate action, zero hunger, and quality education. By reducing the methane emissions from cattle, we decrease the total greenhouse gas emissions, which is in accordance with Goal 13, climate action. Additionally, by increasing animal productivity, we increase the resource efficiency and strengthen food security, contributing to Goal 2, zero hunger. Furthermore, through our educational activities, we spread knowledge about synthetic biology and its potential for sustainable development, which is in line with Goal 4, quality education. Ultimately, our innovation demonstrates how synthetic biology can be used to drive sustainable development.
Addressing methane emission from cows is increasingly relevant, and our project offers valuable insights for future studies. Sustainability is crucial in today's society, and we hope that our efforts will contribute to the ongoing research for a more sustainable future.
Thank you to all our sponsors, especially LTH and LMK, for making our project possible.
Anna! Okay, let's do it on the same day. Let's do it on the same day. 1, 2, 3.